Alright, welcome back. We are looking at the third and last Saturday study session of the microevolution review. And for this one, what I want to look at, let's make this just a little bit larger. We're going to start by looking at the short free response question. It's talking about a team has genetically engineered strain of fruit flies to eliminate errors in DNA replication. Team claims this will eliminate genetic variation. So let's highlight some stuff as we go through here. So we're talking about eliminating genetic variation in engineered flies. The second research team claims that eliminating errors during DNA replication will not entirely eliminate genetic variation. Then we get down to the part down here. So guys, whenever I get to the free response questions, I always like to circle the command word, and we need to provide, I like to square the number to say, so we're just looking for one. We're looking for a piece of evidence that would indicate new genetic variation has occurred. That's for part A. Part B, we're going to be describing, so you cannot just list it, you have to explain it a little bit. Again, we're just looking for one, and this is a mechanism that could lead to genetic variation. So what kind of things cause genetic variation? The third to last one, and again, is another describe. Don't just list something, describe it. How genetic variation in population contributes to the process of evolution. So how does genetic variation help or contribute to the process of evolution in the population? Since this is the play post-it part, I'm going to go ahead and pause the video here. Let you enter in your responses, be thoughtful about it. You don't have to do complete sentences, but give me an outline or some ideas about what you would type if you were really responding to this question on the exam. So assuming you've done that, let's go on and let's look at what the rubric says for this one. So if I go down, let's look at each one. It says right here, evidence for this. How do we see evidence of, and excuse me, I'm going to have to go back because I forgot exactly what our evidence was for. It says provide evidence that indicates genetic variation has taken place. All right, so evidence that genetic variation has taken place, we see new genotype. Let's just highlight. We see new genotypes or phenotypes, or new DNA chromosome differences, or different mRNA sequences or proteins with different amino acid sequences, somehow indicating that there's new genes entered into this population. Part B, we're supposed to identify a mechanism for genetic variation. I tell my students, probably, most likely, you're going to be asked this somewhere on the AP test as well, maybe not in the same format, but it's always good to know the mechanisms for evolution. What causes, genetic variation? So obviously, meiosis during sexual sexual reproduction. I sometimes pronounce it meiosis to not get it confused with mitosis, but it is meiosis. Crossing over of the chromosomes during meiosis, independent assortment of the chromosomes, and then random fertilization. We'll look at each one of those here in just a little bit. You could also talk about emigration, and gene flow, about how virus infection inserts its DNA into the DNA of the genome, and a few other things. Obviously, mutations are here as well. And then for the last one, talking about, and again, I have to go back really quickly to see what we're describing on this one. So if I go here, up again, how genetic variation contributes to the process of evolution. So we are looking at this idea of it's the basis of phenotypic variation, the changing of phenotypes. And remember, we talked about this earlier, natural selection can act on the phenotypes. It does not affect the genotypes, but again, natural selection can act on the phenotypes. So it's good to see that again. Then they're saying here, without genetic variation, there would be no phenotypic differences, which natural selection could not act on then. So that's what they're looking for, but it's always good to go back and review just a couple things. So let's look again at the mechanisms for genetic variation. And one of these is random fertilization. This picture is good because it shows you the hundreds of thousands of sperm that might reach the egg, and each one of those sperm DNA is slightly different. The one that fertilizes the egg will be the one that has the set of DNA that's going to combine with the egg. So that's what the process of random fertilization looks like, that one winning sperm, to say. And then crossing over, this again happens with the prophase 1 of meiosis, and it's the idea of the homologous chromosomes. They're aligned, they cross over, and then they separate, so that this one's just showing you the blue one comes away with a little part of the red one comes away with a little part of the blue. Very important process if you're ever asked about genetic variation. Crossover is an excellent answer. Other sources of genetic variation, we talked about this, independent assortment. It's all about how the chromosomes line up during metaphase 1 of meiosis. And you can see here, if we put all the blues on one side, the reds on the other, we'll get 2 different combinations. And if we crisscross those, so we get those colors mixed up a little bit, again, we'll get 2 separate and different combinations, but it all has to do with how do our chromosomes line up at the very beginning, again, providing some type of genetic variation, independent assortment. Another, some more mechanisms for genetic variation, mutations we talked about. This would be a simple point mutation. We're changing one nucleotide base in here. Then we also have chromosomal mutations. So it can happen on the DNA or on the chromosome. And this one's showing you how a particular gene has been duplicated over here. So it's not just only here, but it's here as well. So mutations can happen on the DNA or on chromosomes. And we also talk about these things. Hopefully everybody's done the transformation lab where you can make the bacteria glow if that's the correct type of plasmid that you received. But the idea here is transformation is good to remember. Another type of one that we commonly talk about, especially with the Hershey-Chase experiment, is transduction, where we get the a virus, a prophage, inserting its DNA into a bacteria. And again, so the DNA from here gets inserted in right here. So another way to mix up genetic variation. Another one is this idea of conjugation. It's another name for bacteria sex. One bacteria is reaching out with its sex pilus and attaching to another one and exchanging DNA or genetic information through here. This isn't truly sex because there's no meiosis involved, but it's a good way to remember what conjugation is. And then we also get transposition or jumping genes located on here as well as a source of genetic variation. Okay, let's look at question number 2. And let's just let it summarize. Actually, let's go to what you're looking at for question number 2, which looks something more like this. All right, a group of botanists studied a variation in leaf mass among tropical trees grown in their natural conditions. The data table summarizes the results for 25 trees in each of the experimental groups. This one says what to do down here. We need to graph. And what are we graphing? The means of leaf per mass area grown in the forest, but we can't forget, and the leaf per mass area in the understory. It also tells us the sample mean should be plus or minus 2 for the the standard error of the mean is what this is, which it tells us right up here what that is. Then it asks us to answer something. It says, based on the sample, so after we graph something, we're going to answer this. And it says, look at these and determine whether there are statistical differences in the trees grown in the forest gaps and the understory. So we'll determine the difference between these trees, and then we'll try to justify or explain our response. I'll give you a few minutes to think about it. Go ahead and try to make a graph here. I'll give you a hint and just tell you we are going to be looking at the at a bar graph. So your x-axis is going to label the 2 major groups down here. You have the forest gaps and the understory. And because you're asked to graph the means, here's your mean, which you'll put on the y-axis. Go ahead and do that, and then we'll talk about the answer. All right, let's look at what the rubric says for this question. First of all, a graph should look something like this. And keep in mind what they're asking you to graph was the mean. So the forest gaps had a mean of 4. Understory had a mean of 2. And other thing we should look at is if I go back to this section right here and it shows us the standard deviation. We didn't really have to do anything with that, but they use that to calculate the standard error of the mean. And basically the standard error of the mean, these are bars that are going to describe the accuracy of the data that was used to make the graph. And you can kind of get an idea. It's going to be somewhat related to standard deviation, where this one is greater. It's more standard deviation means it's more spread out, and so we'd expect to have a greater standard error of the mean down here. It does say on the equations and formula sheet that students don't necessarily have to be able to calculate the standard error of the mean, but they should know what it means. And again, what I want you to know that these are the error bars that are going to show how good the data is or how good it's not. So let's go back to the rubric really fast, and you can see they graph the mean right here, and then these bars right here are the standard error of the mean. And so this one started at 4, and it went up to 0.5. It also went down by 0.5. So it's basically saying it could be off by this much. Versus this one over here, here's the mean. It started at 2, went up to 3, and went all the way down here to 1. So it's off by 1 direction this way, or 1 unit this way as well. So this one has a greater, we could look at this and say the data isn't as good in this one. So that's how we'd graph the standard error of the mean. Let's go ahead and look at the question really quick, or the rubric for this. You get points for correctly labeling, graphing, and the proper units on the graph. The bar graph has the correct means on it, and you have the correct standard error of the means on those as well. Those are the bars that show you how good your data is. Down here for identifying, we could say, yes, the two populations are statistically different. And what would you use to justify that? You would say, well, the error bars, or you could talk about how you're 90% confident the intervals between the two populations do not overlap. I think a better idea here is to say the error bars were slightly off. One was larger than the other. So let's go into the long free response. And for the long free response, I'm going to have to pull this. All right, let's keep going with this. The long free response, we're looking at this. And I'll let you look through this, but it's basically another Hardy-Weinberg question, and it's asking you to calculate the frequency of the homozygous dominant, the heterozygous, and the homozygous recessive. It wants you to discuss how the Hardy-Weinberg principle of genetic equilibrium can be used to determine whether the population is evolving, and then to identify a particular environmental change and describe how it might alter the frequencies. This one, again, this one's a discuss, but this one's an identify. So it should be pretty easy, but then we have to explain which Hardy-Weinberg principle would not be met. I have about 1.5 minutes left, so I'm going to try to be fairly fast. On this one, what I'd encourage you to do is just make sure, again, you solve for mind your P's and Q. And this one tells you right here the recessive allele is 0.4. So you could come up here and say Q is 0.4. You've already solved for that. P down here is 0.6 is equal to P. So if you wanted to get the homozygous recessive, you would say, well, that's just going to be P squared. You would take your 0.4 and you would square it to get your answer there. Then 2PQ, you do just plug and chug, and Q squared, you do the same thing. So I'm going to go ahead and pause it right here so we have time to look at the rubric really quick, and we'll go ahead and do that. All right, I'm just showing you the rubric now for question 3. Hopefully you were able to solve for these pretty easily. I don't have a whole lot of time in this video, but this gives you an idea. I'm just going to scroll down really quickly so you get an idea of what the rest of this looks like. And you can pause those and look at those to double check your answers. I hope that was helpful, and hopefully you have a good idea of microevolution.